Thanks, Katie, and to the other organizers for the opportunity to speak here. I've learned a lot so far. The work I'm going to talk about today doesn't actually involve any analysis of Neanderthal DNA or other archaic DNA, but it sort of builds upon work that has been really exciting work that's been done on that area and uses more population genetic theory and simulations to explore consequences of this introgression that we can't immediately get from the data at this point. So this is work I did at the end of my Berkeley PhD with Rasmus Nielsen. And just to set the stage, before there had been any sequencing of archaic DNA, there was a controversy in human genetics regarding the origins of the human species, where there was a lot of support in the anthropology committee for this multi-regional hypothesis, where we know that there were Homo erectus and other archaic hominids all over Europe and Asia, as well as Africa. But the ultimate origin of hominin lineages within Africa. And so some people believed that humans in Europe and Asia resulted from this sort of continuous mixing of modern human traits coming out of Africa, but then mingling with more locally adapted traits that originated in other parts of the world. And in contrast, there's this out-of-Africa replacement hypothesis, which has a modern human lineage completely replacing archaic hominids who dispersed to other regions. And so when we first started to get mitochondrial DNA from humans and from Neanderthals, it seemed to really support the replacement hypothesis, where you have Neanderthal mitochondrial variation lying completely outside of modern human variation, which is rooted within Africa. But a bit more murky picture emerged after the sequencing of a draft nuclear Neanderthal genome, which found slightly more similarity between non-Africans and Neanderthals than between modern Africans and Neanderthals. And so now the leading theory is this leaky replacement hypothesis, where a very small amount of human DNA outside Africa appears to be derived from archaic admixture. So we think about 2% of modern human DNA outside Africa has an origin in relatively recent Neanderthal gene flow. But the question that Rasmus and I wanted to ask was, does that really mean that 2% of our ancestors were Neanderthals? So this might not seem like that complicated a question, but the main complication, as we see it now, is that there is a lot of evidence of natural selection against Neanderthal DNA in human populations. Of course, there's also evidence of selection for Neanderthal DNA, but so far that looks more localized. And actually, if you look at genome-wide, so there was an exciting paper a couple years ago from Sriram Sankaraman and David Reich, where they looked at the average Neanderthal admixture fraction over the genome as a function of the B statistic. B stands for background selection, and basically regions of low B statistics are very close to genes and other regions that appear to be highly conserved throughout evolution, and regions of high B statistic are believed to be more like junk DNA that is not under strong purifying selection. And so the pattern from their paper here is that regions under strong evolutionary constraint only on average have about a quarter as much Neanderthal DNA as regions under weak constraint. And so one thing that could be behind this that they talked about in their paper are Dobzhansky-Muller incompatibilities. So that's this idea where, when you have an interaction between two loci of some kind, and then you have two populations that stop interbreeding, then through selection or drift in one population, you could have the emergence of an allele at one locus that could interact negatively with certain alleles at the other locus. And So evolution will favor the exclusion of one of these alleles, basically, or one of allele combinations that interact negatively with each other. But you could have an emergence of a negative interaction between species so that if they come back together, they might not be completely fertile. And we know this is a very important process in speciation in general, but one problem is that a whole lot of these Dubzhansky-Muller incompatibilities would be required to create a pattern like in the B-statistic graph I showed you. Because each one requires two substitutions that have some kind of function, and there's not much divergence between Neanderthal and human sequences. So we wanted to see if there could be a more parsimonious explanation for this pattern. And the thing that we looked at was that Neanderthals have been shown to have a much lower genetic diversity than humans. They appear to have a very small effective population size. And so from some theory by Tomoko Oda, this is from a 92 paper, but much earlier, basically the fixation probability of a new allele depends on the product between population size and the selection coefficient of an allele. And so if you have a very weakly deleterious variant, then if humans had a 10 times larger population size than Neanderthals did, then you can basically have a much higher probability for that allele to fix in Neanderthals than humans. And then once that allele gets into the human population, you have the potential for it and the surrounding Neanderthal DNA to be strongly selected against. So we did some simulations using a forward simulator by Philip Messer, who used to be here, where you have weakly deleterious alleles fixed within a Neanderthal population, and none of these in humans. And basically, if you have these alleles spaced out pretty widely in the genome, you get strong selection against Neanderthal DNA throughout the first 20 or so generations of interbreeding. But then once everyone has almost the same amount of Neanderthal DNA, the selection slows down. And then what you get is much slower selection that occurs in the vicinity of the deleterious variants. And you get a gradient of less Neanderthal DNA near the deleterious mutations and more far away from them. And we actually showed that doing a simulation of mutation accumulation within a realistic joint human Neanderthal demography, that these weakly deleterious mutations do appear to accumulate in the Neanderthal lineage. Basically, you get to a point where the median fitness of Neanderthals only appears to be about 60% of the fitness of the humans after they accumulate mutations with a small population size for a long time. So this is sort of showing the distribution of fitnesses within populations, but the means obviously being very different. And if you separate this fitness differential, partition it into loads of mutations of different selection coefficients, you see that most of the difference between humans and Neanderthals is due to weakly deleterious variants with selection coefficient around maybe a selective disadvantage of 1% to 0.1% per generation. And you actually get a very similar result if I've been talking about additive mutations so far, that where a heterozygote is half as bad as a homozygous variant is, but you actually get a similar load if you have different dominance coefficients, like recessive ones. This is a graph similar to what I showed you with my simpler simulation at the beginning, where if you actually need to start with a population of around 10% Neanderthals to reach an equilibrium Neanderthal percentage of about 2%, like we see. And so I think that's really interesting because 10 to 1 is about the ratio of the effective population sizes of the two species. And so it suggests maybe you could have even had random mating between these species, which after this selection occurs, looks more like leaky replacement of a much smaller Neanderthal contribution. Interestingly, a different picture happens if most of the mutations are recessive, where you actually appear to get positive selection for Neanderthal DNA just because humans and Neanderthals would have had different damaging variants that get masked. And so this is an example of dominance heterosis, which is known to occur in quite a few species, like these, especially crops, like is shown here. And so just to recap, but if most mutations were additive or dominant, we would have had selection against Neanderthal DNA explaining why you observe few of these variants in near genes. And just want to talk briefly what the impact of that could have been on human fitness. So there's been a lot of debate about whether smaller bottlenecks outside of Africa could have impacted the deleterious mutation load, made it higher outside of Africa than within. And basically, we did some simulations indicating that Neanderthal admixture could have had an impact around the same order of magnitude as the out-of-Africa bottleneck on human fitness. Yeah, I'll just wrap up now. These simulations predictions extend to any case where you have secondary contact between diverging populations that have different effective sizes, which could be important in conservation biology, since just with species moving their ranges a lot these days, you could potentially have a lot of human Neanderthal contact type scenarios that could impact the fitness of a number of species. So this could be good for small inbred populations, but bad for healthier outbred ones. None of this is saying that Dobzhansky-Muller incompatibilities couldn't have existed between humans and Neanderthals, especially in these regions that appear to be almost devoid of Neanderthal DNA, but we also don't need to invoke them to explain these patterns of pervasive selection that have been observed. And yeah, I'd just like to thank Rasmus and then all the people in my old and new labs and elsewhere who've helped me talking about this project.
Any questions for Dr. Harris? Okay, right here in the front.
Hey, two questions. One is, I guess, the flip side of what you showed in the model is that we expect in the Neanderthals, admixture from modern humans into Neanderthals to be actually adaptive, right? And I wonder if you're aware of evidence for that. The second question is, I wonder if the comparison of autosomes and X diversity under this model should show some difference between Muller-Dobzhansky incompatibilities to just a difference in mean fitness in populations.
Yeah, those are both very interesting questions. Just answering the second one first. So there's overall a lot less Neanderthal admixture on the X chromosome compared to the autosomes. And so that's consistent with the idea that dominance effects are very important in the dynamics because since males only have one X chromosome, all deleterious mutations must be dominant in them. And so even if a modest number have recessive effects, then if those become dominant in males, you would predict lower admixture on the X. So there's a paper by Graham Coop drawing similar conclusions to mine with different methods where they looked at the X admixture in a bit more detail. They sort of suggested that you would need some sex-biased admixture too to explain what you say, but yeah, I guess that's definitely an avenue for future work. As far as whether human admixture was beneficial to Neanderthals, I think yeah, the problem is we just have so few Neanderthal sequences. It would be helpful to have more high-quality Neanderthals to be able to see evidence of that kind of selection. But hopefully we'll get more of those soon as well.
One more question? Okay. Dr. Feldman?
In your result, you had a 10-to-1 difference between Neanderthals population size, between modern human population size and Neanderthal, right? It was 10-to-1. Yet when the first bottleneck is occurring out of Africa, and maybe the first contact between a propagule of modern humans contacts Neanderthals, wouldn't it be the other way around, that the population size of Neanderthals will be quite a bit bigger than the invading population of modern humans? And what would be the consequence of that?
So I think that's definitely possible. So I think it takes quite a while for a population bottleneck to affect the deleterious mutation or the distribution of selection coefficients of the segregating mutations. So I've actually done simulations like this under the scenario of a shorter bottleneck to look at if it could be important in polar bears. And with that, when you had a shorter bottleneck, you see much less of these effects. So I think it's very important for the size difference to have been maintained for a long time. But where the effective size of the humans who met Neanderthals could matter a lot, I think, is the variance of admixture proportions among the early hybrids. And that could be important for the size of these deserts, where you could get some region that's devoid of Neanderthal ancestry without any selection more easily if the original population of hybrids was a lot smaller than the long-term effective size.
Awesome. Thanks so much, Dr. Harris. Warm applause.